If you just want to know the chemical structure of the protein product emerging from translation, the genetic code is sufficient to model the system. However, often we want to know quantitatively how much of that protein will get made in the cell at any point in time, and for this we must somehow model translation as a process. This turns out to be quite challenging, but let's start with the most basic model. Suppose we have a droplet of water containing a protein. We can describe the number of molecules of that species as a variable p, and suppose that at some initial time point p has the value p0 equals 3 protein molecules. A natural value would be in the range of 1 to 100,000 molecules of p in one cell. If this is truly just protein in water, then the only thing that can happen is that protein could get degraded. There are all sorts of mechanisms by which a protein can degrade. The peptide bond could spontaneously hydrolyze by reaction with water. UV light could result in photochemical damage, and it might spontaneously unfold or aggregate. In a real cell, under rapid growth conditions, the doubling time of the cell usually dominates any active degradation process. But keep in mind that there are many distinct mechanisms involved in the degradation of a protein in the cell, but they're usually lumped together into one term that is assumed to be first order with respect to protein concentration. Thus, we describe the change in p over time as a simple first-order differential equation. That can be solved for p as a function of time as being the initial concentration of p times e to the negative gamma t. If we plot that, we see the classic and simple exponential decay behavior where p asymptotically goes to 0 over time. Similarly, we can model the rate of translation according to simple first-order kinetics. The change in p as a function of time is the sum of two processes, synthesis and degradation. The synthesis term is proportional to the mRNA concentration, and the degradation term is proportional to the amount of protein present. If the system achieves a steady state, then the protein concentrations no longer change, and the rate of change of p becomes 0. So we set this equation equal to 0, and then we can solve for the concentration of p at this steady state by just doing some simple algebra. And we find that p is equal to beta m over gamma. This very simple math turns out to be a good model of constitutively expressed proteins under vegetative growth conditions. The reason for this is that degradation rates are dominated by growth of the cells and dilution of the cytoplasm, and this rate is on similar time scale to the rate of protein synthesis. However, most of the detail is hidden under this term beta that makes it difficult to relate this math back to the sequence. In a real-world situation, we are trying to express some protein and hit some specific expression level in the cell. We have three easy dials we can turn to alter the expression. We can change the promoter, the RBS, or the codon usage of the gene. We can write this differential equation models to describe the system incorporating any detail we want, and from this we can do computer simulations that will tell us what each system will do. The challenge here is that we need to map the parameters in the model to physical parameters and then relate those parameters to the sequence in order to choose the specific sequences that will display the behavior of that model. In the transcription chapter, we talked about how transcription rates can be related to binding constants for sigma 70 and various other terms. We saw that the sequence of the promoter is locally encoded, and thus promoter activity tends to be insulated from downstream and upstream sequences. It was fairly easy to describe promoters as parts and stamp a relative expression level on each promoter part that predicts its relative strength. A similar approach can be tried for ribosome binding sites. RBS parts can be generated in much the same way that promoter parts are generated. You start with a composition of promoter, RBS, coding sequence, and terminator, then apply saturation mutagenesis to the RBS region. If your CDS part is readily assayable, then you can pick colonies with distinct expression levels, sequence this, and from them have a set of RBS parts. The test for the modularity of these parts is done by putting the family of RBS parts in different contexts. Here we have a family of 7 RBS parts placed upstream of either RFP in red or LAC-Z in blue. All constructs have the same promoter and terminator, they just differ by the choice of ORF and choice of RBS part. Notice that the rank order of the ribosome binding sites is different for the two proteins. For perhaps half of them, which correspond to the weaker RBSs, the ratios of the different activity levels seem fairly proportionate, but at the higher levels things get quite out of whack. This very simple method of encapsulation turns out to have some validity. Perhaps 50% of the variability is accounted for. However, it is clear that the junction between the RBS and the coding sequence for simple ribosome binding sites is not trivially modular. Before we move on, let me make clear that we are discussing translation in prokaryotes here, and the sensitivity of the 5' UTR is far less pronounced in eukaryotes. In yeast, it turns out that tuning ribosome binding sites has much less of an effect on gene expression.